Good morning, I'm Bianca Barzaghini, a PhD student in bioengineering at Politecnico di Milano in Italy. I want to thank the scientific committee for giving me this opportunity, and I will introduce my project that is focused on 3D scaffold named Nichoid, which is optimized for human adipose-derived stem cells, in order to develop more efficient stem cell therapies. In particular, my work is focused on spinal cord injuries, which causes temporary or permanent loss of function, of mobility, and feelings. Unfortunately, spinal cord injury affects 9 million of people in the entire world with a cost of $100,000 per patient. Currently, there is no cure for spinal cord injury, and the patients are supported with therapies to limit pain and with technologies as exoskeleton to support the limitation of paraplegia and tetraplegia. But in the recent year, a new approach is becoming more relevant, and regenerative medicine and in particular stem cell therapies. This approach is focused on possible recovery that gives new hope to the patient. Several research groups study embryonic stem cells and induced pluripotent cells in neurodegenerative disease. Despite interesting results in the animal model, the transition to clinical phase in human is difficult due to the risk of teratoma and tumor formation and the use of chemical agents to avoid spontaneous differentiation. Indeed, from 142 initial clinical trials, only 2% reached the phase 3. Our approach is totally different. We want to improve the beneficial effect of autologous stem cells, and the cells are harvested directly from an invasive liposuction of subcutaneous adipose tissue of the patient, expanded in vitro in our scaffold, so in a 3D environment, to obtain a consistent number of cells to guarantee a therapeutic effect, and then re-implanted in the same patient, limiting the immune acute response. In 2012, Professor Raimondi started to study a 3D micro-scaffold named Nichoid, which mimics the microarchitecture of stem cell niche. The goal of this scaffold is to maintain or even increase the stem cell potential and ensure a better therapeutic effect. Nichoids are fabricated by two-photon laser polymerization using a photosensitive and biocompatible resin. And this engineered niche has a precise geometry of the pore and it's optically accessible. Thanks to the isotropic mechanical stimuli in feeder-free cultural conditions without the use of chemical factors, the niche permits to maintain a pluripotent state of the cells growing inside, in particular with embryonic, neural, and rat mesenchymal stem cells. After the fabrication, I expanded the human mesenchymal stem cells inside the graduated pore nichoids, but unfortunately, several cell detachment protocols did not allow a proper cell detachment, and 90% of the cells were trapped inside the small pores. This is a crucial step to collect cells for a future transplantation and so require a further investigation. The solution is to enlarge the structure of the grid. A new niche called cubic niche is therefore designed, whose elementary pore measure 30 by 30 micron and 60 micron in height, which are comparable to the dimension of these human cells. To investigate the biomechanical stimuli of the cells, we develop a computational model of the structural interaction between cells and the scaffold pores using constant multiphysics. So we consider an elastic material with a reported parameter and the worst scenario caused by a force of 70 nanonewton for each focal adhesion from literature data. We calculate a displacement of only 2 micron that is comparable to the first initial structure. So in this case, the structural stability is maintained, and we can perform further experimental validation. Here I report the same images of the samples, and thanks to the fast two-photon polymerization process, we can create scaffolds in only 2 hours of fabrication. The single block is repeated on the glass cover slip with an area of 80 mm in diameter, and before the use, I sterilized the samples with just ethanol and UV exposition. In the first step of biological validation, I grew human adipose-derived stem cells for 7 days in standard condition and inside the new cubic nichoids. In these immunofluorescence images, the red marker represents vimentin, which is the major cytoskeletal component of the mesenchymal stem cells, and we can observe a difference in morphology. In flat standard condition, the nuclei are elongated, while in the 3D reconstruction, the nuclei maintain a spherical shape. I then evaluated the nuclear aspect ratios, the ratio between the minor and the major axis of the nucleus, and I obtained a value close to 1 in the cubic nichoid, indicated a significantly more spherical geometry. This confirms the hypothesis that the cubic pore nichoid induce an isotropic cytoskeletal tension. From literature analysis, a spherical nucleus corresponds to a pluripotent state, and furthermore, as reported from Hamak et al., the activation of the marker octamer4 and the consequent SOX2 and NANOG is also connected to an undifferentiated state. Both these aspects were present in my experiment, and the measure of these stem cell markers in cubic nichoid results upregulated. This evidence shows how an only mechanical isotropic stimuli from the niche could induce a pluripotent states in human mesenchymal cells. As cells are pluripotent, they are able to differentiate better and respond more effectively to induced external stimuli. Since the study dealt with spinal cord injury, I decided to differentiate the cells into the neural commitment. At first, the cells were expanded in the cubic niche, effectively detached, collected, and then differentiated to simulate inside an in vitro culture what could happen after cell transplantation in vivo. Then I marked the cells with beta-tubulin 3, shown in green, which is a neural marker for cytoskeleton, and the expression is significantly higher in the cells previously expanded in the niche with respect to the flat control. This evidence suggests a better plasticity of these cells in response to the external stimuli. To sum up, we optimized the structure of cubic pore nichoid and we performed the consequent validation with biomolecular and confocal microscopy approach. We can assert that this new geometry is suitable for the expansion and characterization of human mesenchymal cells, and in future we want to perform a deeper analysis of the entire genome of human mesenchymal cells and begin to set the protocol for stem cell transplantation in a preclinical animal model of spinal cord injury. I want to thank my professor and my lab teams to support me in this challenging project, and thank you for your kind attention. And I'm glad to answer all of your questions.